There was a long and fairly heated debate on the true nature of light. For example, experiments like the interference experiment and diffraction effects showed that light had to be a wave phenomenon since only waves experience these types of phenomena. On the other hand, there were also experiments happening which had results which were clashing with the idea that light is a wave. One of these experiments revolved around the photoelectric effect. So let us understand what kind of setup they used to come to the conclusion that the wave theory cannot fully explain the behavior of light. So here we have a simplified schematic of the experiment for the photoelectric effect. What we have here is an evacuated glass tube. So here it is an evacuated glass tube, meaning that we have a vacuum inside this tube. This makes it easier for electrons to move from one point to the other without suffering collisions from gas molecules. There is a small optical window placed here, as you can see, and this allows us to send light of various frequencies and various intensities as required by the experiment at different periods of time. So we can change the intensity of the light or the brightness of the light. We can also change the frequency or color of the light. So if I'm sending in red light, that has a different frequency. If I'm sending in green light, that has a different frequency, and I can change the brightness of the light or the intensity of the light as well. Now, this incident light is made to hit a plate of photoelectric material, and this plate is called our emitter. So here we have an emitter plate. This will emit photoelectrons when light of some intensity is hitting the plate. And these electrons can move in different directions. But if we provide it a positive potential to move towards, for example, if we take this plate, which is our collector plate. So this is our collector plate, and we place it at a positive potential with respect to the emitter plate, we will see the movement of electrons from one side to the other. So here we have electrons. These are photoelectrons moving to positive potential under the influence of the collector plate. Now, we can reverse the direction of the potential being placed across the plates. In other words, I can make the collector positive or negative with respect to the emitter plate. This is accomplished using a device called a commutator, which is what we see here. So this is our commutator. By changing the direction of the connections, I am changing the direction of potential within this device. Now, we can also change the amount of potential by using this variable resistance. So this is our variable resistance, and we can use it to decide what is the magnitude, the amount of voltage being placed across these plates. In addition, we have a voltmeter, as you can see, and we have a microammeter because the photocurrent is very small, so we need a very sensitive device to be able to measure this current. Now let us see how this device is going to function. Now, to start the experiment, we will place a light source in front of the optical window. The light source will send a measured amount of light onto the emitter plate. The electrons in the emitter plate will gain this energy and become excited, and some of these electrons will leave the plate and start moving in the space between the emitter and the collector plates. By ensuring that the collector is at a positive potential, we can ensure that these electrons start moving towards the collector. Now let's see what happens. As these electrons start moving towards the collector plate, they are feeling the positive potential of the collector. Now let's say that we have a fixed intensity of light, and we can gradually change the collector potential so it becomes more and more positive. What will happen? If you observe the reading on the microammeter, you will notice that for a set value of intensity, that is for a fixed value of light brightness, the photocurrent will increase up to a particular level and then stop increasing, which means that no matter how much you increase the voltage difference between the emitter and the collector, beyond a particular point, the photocurrent will stop increasing further, irrespective of the voltage between the emitter and the collector. This value will remain fixed as long as the brightness or intensity of the light is fixed, and this is what we call a saturation current. Saturation current means that all the photoelectrons being emitted from the emitter are reaching the collector, so a further increase in potential will not increase the number of electrons, and as a result, the photocurrent becomes constant or saturated. Another interesting thing to notice here is that if we keep on increasing the intensity of light by a set amount, we will see that there is a photocurrent increase also by a set amount, and this relationship remains constant. In other words, if I were to draw a graph between the intensity of the incident light, the brightness of the incident light, and the amount of photocurrent, I would find that it is a straight line, because the relationship between the photocurrent and the intensity of the light is constant for a given amount of potential. In other words, the photocurrent will increase linearly with the intensity of light. For the second part of this experiment, now we will reverse the direction of the voltages. Now, for the next part of the experiment, I will use the commutator, that is this device here, to reverse the direction of the voltage. So now the positive terminal becomes negative and the negative terminal becomes positive. In other words, I am making the collector negative with respect to the emitter. So if I put a negative potential on the collector, what will happen? The electrons will sense this negative potential and that will prevent them from reaching the collector. A few of the electrons will have enough energy to reach the collector despite this, but a large fraction of the electrons will be unable to reach the collector. I can keep on increasing this negative voltage until no electron emitted from the emitter, no photoelectron is able to reach the collector. This negative potential, at which there will be no detectable photocurrent inside the apparatus, is known as the stopping potential, which is given by the symbol V0. This is the reverse voltage at which no photoelectron emitted from the emitter is able to reach the collector, irrespective of the intensity of the light we are using. For a constant frequency of light, we will notice that the stopping potential remains the same no matter how intense the light becomes. So what we are noticing here is that the stopping potential has no relation with the intensity of the light, but it does control the amount of photocurrent inside the apparatus. Now, to take the experiment further, we can change the frequency of the incident light inside this apparatus. That means that we are using light of a different color. For example, if we were so far using red light, we can replace that red light with a violet light or a blue light of the same intensity as before. So now we are not changing the brightness of the light, but we are changing the color of the light or the frequency of the light. And what do we observe? We will see that if we increase the incident frequency of light, there will again be a photocurrent detectable in the apparatus, even though we are applying a stopping potential. This means that the existing stopping potential for the previous frequency of light is no longer enough to stop the electrons from reaching the collector. In other words, we see that the stopping potential has only to do with the frequency or the color of the light and has nothing to do with the intensity of the light. Thus, while we see that the intensity of light and photocurrent vary linearly with each other, we also see that the intensity and stopping potential are independent. On the other hand, we also see that the stopping potential is related linearly with frequency. If you keep on increasing the frequency of the light, we see that we have to keep on increasing the stopping potential. In other words, in this experiment, the amount of photocurrent available to us is determined not by the frequency but by the intensity or brightness of the light. On the other hand, the stopping potential that we require to prevent the photocurrent from conducting from the emitter to the collector is determined only by the frequency or the color of the light. The intensity or brightness has nothing to do with the stopping potential. This result, which says that the photocurrent depends only on intensity and the stopping potential depends only on the frequency, does not really mesh with the idea that light behaves as a wave. This is because waves are a continuous phenomena. A surface, if it is absorbing light as a wave, is absorbing energy continuously. And that energy absorption will take place for any intensity of light and any frequency of light. Here, we see that there is no photocurrent emission below a particular threshold frequency for a given stopping voltage. Which means that it doesn't matter how bright we make the light, it doesn't matter how much energy we are putting on the surface, there is no absorption of energy and no photoelectronic emission. We will now understand how we can explain these results, and the insight into the behavior of light was provided by none other than Albert Einstein in his famous photoelectric equation.
Manorama Horizon. Learning made simple.